I'm Prevotella. I'm a third year biochemistry student at the University of Liverpool. On my research project, I've been working alongside Dr. Chris Hope, who's a lecturer in oral biology as well as a senior researcher in the dental department. We've been working together to identify if 505 nanometer light can be used to kill this black pigmented anaerobic bacteria called Prevotella Intermedia, and this is because this bacteria is involved in the initiation and progression of periodontal disease. And this bacteria is photosensitive because it contains this iron-free portofulfin-9 ring, and also because this bacteria is anaerobic and very sensitive to oxygen. The entire experiment has been carried out in an anaerobic chamber. So we first grew this bacteria on 5% defibrinated horse blood agar plate, and we left this agar plate to be incubated in this anaerobic chamber for 24 hours. And then once this Prevotella intermedia bacteria had grown, we created bacteria suspension using PBS buffer, and we measured the optical density of this suspension at 650 nanometers using a spectrophotometer to obtain an optical density of 0.1. And once this optical density was obtained, we transferred 200 microliters of this solution in a 96 well plate and then exposed the 505 nanometer light to this bacteria suspension for different ranges of time. For example, we used 10 seconds, 30 seconds and 60 seconds. And then once this bacteria suspension had been exposed to this light, we transferred it, we carried serial dilutions on it and transferred the corresponding dilution to its corresponding agar plates, and then we left these agar plates for incubation, and then we counted the colonies after 1 or 2 days using a colony counter. And once the experiment had been carried out, we carried out statistical analysis using Microsoft Excel to compare and to calculate the percentage kill at each time, so the 10, 30 and 60 seconds, and it has been shown to be successful, as because at 10 seconds we were able to obtain 89% kill, but at 60 seconds we obtained 98% kill. And so it has been successful, as the more exposure to the bacteria using the 505-9 light, the better it kills. And throughout this experiment, I've thoroughly enjoyed working on it because I've not felt like a student, I've actually felt like a researcher. And also my supervisor has always been very supportive and provided me with any help that I've needed. Thank you.